Primary structure. The sequence of amino acids in a protein is known as primary structure. The specific information that determines the final three-dimensional form adapted by a protein is inherent in its primary structure. That is why it is very important they are having a definite amino acid sequence, then they are going to be a perfect protein. The many genetic diseases result in proteins with abnormal amino acid sequences causing improper folding and loss or impairment of normal function. The peptide bond, that is amide linkage, covalent bonding, and not broken by conditions that denature the protein. Here you see primary sequence. There are amino acids are in definite sequence, amino acid and the peptide bond. You must be knowing their amino acids are modified in different way, such as phosphorylation, acetylation, hydroxylation, methylation, disulfide cross-linking, and carboxylation in different molecules and different proteins in our body. Phosphorylation, that is addition of phosphate, is done on serine, threonine, and tyrosine. Acetylation, addition of acetyl groups to lysine residues and amino termini. This hydroxylation happens as the addition of an OH group to a proline or lysine. Disulfide cross-linking is done in two cysteine residues, and that forms the disulfide linkages. Methylation, that is addition of methyl groups to lysine or arginine. And carboxylation, that is addition of carboxylic acid group to glutamine to make gamma carboxyglutamate. These are the modifications of certain amino acids for their to perform a definite function in our body. Now the secondary structure, that is the local spatial arrangement of a polypeptide backbone atoms, and examples are alpha helix, beta sheet, beta band. This peptide bond is partial double bond and they are in trans form, and due to partial double bond the rotation about this bond is prevented. Thus conformation of peptide backbone is constrained and having regional bends and local folding. You see here is the alpha helix and this beta sheets having 3 strands. There are 3 covalent bonds in a polypeptide backbone, but the free rotation is only possible around the 2 of the 3 covalent bonds. These are alpha carbon to the carbonyl carbon bond, that is the psi angle, and alpha carbon to nitrogen bond, that is the high angle, and these 2 single bonds show free rotation. You see here there is the peptide bond, 2 peptide bonds, but the rotation is only on the psi angle and the high angle. Here you can see the rotation is only on the alpha carbon, which is attached to the carbonyl carbon and amide nitrogen. This freedom of rotation about 2 bonds of each amino acid allows protein to fold in many different ways, and this psi and high angles determines the path of polypeptide chain. And regions of ordered secondary structure arise when series of amino acid residues adopt similar psi and high angles. There is one Ramchandran plot. There was a scientist, Gopal Samundram Ramchandran, recognized that many combinations are forbidden. They are not possible, and he visualized on a 2-dimensional plot called Ramchandran plot. You see here the beta sheet is in the upper left corner and alpha helix on the lower left corner, and left-handed helix is really not possible, and the other conformations are not feasible or practicable. Alpha helix. This is a rigid, tightly packed, right-handed spiral structure, and that is the backbone core. And the side chains of the component L-amino acids, extending outwards from the central axis to avoid interfering sterically with each other, such as keratin, which is a fibrous protein, and myoglobin, which is a globular protein. And these polypeptide backbones of alpha helix are twisted in equal amount of high angle, that is minus 57 degree, and psi angle of approximately 47 degree. A complete turn of the alpha helix contains an average of 3.6 amino acyl residues, and the distance per turn is 0.54 micrometer, and distance between each amino acid residue is 1.5 Armstrong. And these are stabilized by hydrogen bonds between the NH and CO group of main chain. And hydrogen bonds are individually weak but collectively serve to stabilize the helix. And this hydrogen bond is maintained apart 4 residues ahead in the sequence. And this maximum number of hydrogen bonds and van der Waals interactions make them thermodynamically feasible for the formation of an alpha helix. And these right-handed helices are energetically more favorable due to proteins contain only L-amino acid, and their side chains don't interfere in the structure. You see, there is the hydrogen bond and the amino acid, 4 amino acid ahead of the particular amino acid, and they are forming hydrogen bonding. Here you see hydrogen bond. The amino acids which disrupt an alpha helix: proline having secondary amino group, glycine, small size, charged amino acids, glutamate, aspartate, histidine, lysine or arginine, they electrostatically repelling each other or forming ionic bonds. And the amino acids which are having bulky side chains such as tryptophan, valine, isoleucine. Now the amphipathic helices. There are hydrophobic R groups on one side and hydrophilic ones on the other side, so that they form interfaces between polar and non-polar regions and create a channel up or a pore that permits specific polar molecules to pass through the hydrophobic cell membranes. Now the beta sheets. These are of 2 or more polypeptide chains in the extended formation, zigzag or pleated pattern, and the R groups of the residues point in opposite direction. And the distance between amino acids is 3.5 Armstrongs, and they are visualized as broad arrows. And what are the stabilizing forces? The hydrogen bonds, and that is interchain hydrogen bonds. You see in the alpha helix, and this is beta pleated sheet. Here direction of C terminal and direction of C terminal, they are anti-parallel beta sheets. Parallel and anti-parallel sheets separate polypeptide chains or segment, and they may be in anti-parallel form or parallel to each other. In the globular proteins, beta sheets always have a right-handed twist, and the clusters of twisted beta sheets form the core of many globular proteins. Now the comparison of a alpha helix and beta pleated sheets. Alpha helix, hydrogen bonds, intrachain between N and N+4 residues and parallel to helix axis. In beta sheets, it is interchain and perpendicular to the chain axis. residues, small and uncharged residues, and